So my name is Sarah Parrott. I'm the Associate Director at the Faraday Institute, and I'm very glad to introduce our speaker for today, who is Bridget Bannerman, who'll be talking to us about enhancing diversity in clinical trials, a touch of humanity. Dr. Bridget Bannerman works with the Alliance for Cancer Early Detection in Cambridge, where she's a member of the Operations Board and the Programme Manager, as well as serving as a member of the Early Cancer Institute Steering Committee. She's also a research professor at the University of Sierra Leone, piloting initiatives in global health and digital education. Dr. Bannerman specialises in evolutionary and theoretical systems biology, having obtained her PhD in biochemistry from the University of Cambridge and an MSc in informatics from the University of Manchester. Bridget has had a rich and varied career in drug discovery, clinical trials, and research management, working within Cambridge University Hospitals, the Medical Research Council, and the Cambridge University Departments of Biochemistry and Medicine, as well as the Wellcome Trust Sanger Institute. Over the years, she's led institutional projects sponsored by Pfizer and other pharmaceutical companies, as well as the Royal Society and the WHO. As a champion of diversity in clinical trials, Dr. Bannerman has spearheaded patient engagement and recruitment strategies across diverse populations in Africa, Europe, and the Americas. So you can see she's very well qualified to speak on this topic today. So welcome, Bridget.
Thank you very much, Sarah, and thank you all for being here and for having me to talk about this topic, enhancing diversity in clinical trials with a touch of humanity. So talking about clinical trials, I'm looking at it from two broad perspectives, as scientific necessity and also as an ethical obligation. Scientifically, we are examining how genetic, environmental, and socioeconomic factors impact on disease progression and treatment efficacy across different populations. Ethically, we are looking at how marginalized populations benefit from medical research and how we can have equitable access to the benefits across different populations. I've also witnessed how my own faith has supported my roles, the diverse roles I have taken over the years in terms of helping out in diversity. The United Nations SDG 3 goal talks about wellness, health, and well-being for all. So the question we are exploring here today is, what are these barriers we encounter and how are we overcoming them, and how does faith play a role in overcoming them? So I will take you through a journey where we examine this topic of diversity in clinical trials under 4 different categories. So public health has same goal to ensure medical treatments and are safe and effective for all populations. And I have here the concept of homeostasis, which is the body's ability to maintain stability despite external and internal changes. The body is in a constant flux. The biochemical processes occurring all the time in the body. And when there is imbalance, the disease is present. And the body is constantly fighting to maintain a healthy balance. In the same way, in the physical world, there is order, there's chaos. And we're always fighting to always achieve a balance. And different balance for one person might be different for what balance for another person means. Similarly, countries, cultures, continents, balance means different things to different people. Now, how are we navigating all of this? Because despite all the differences we say, we are all at the core, we are all human beings and we have similar needs, and health is one of them. So how we, in a sensitive manner, navigate these balances as we approach different people, different cultures, it's important. And so when I'm talking about the concept of homeostasis, I'm not talking about it just from a biological principle, but how it can play a role towards designing inclusive, ethical, and effective clinical trials. The evolutionary perspective will come more on a scientific basis, because here we're looking at how both biological and environmental factors, how we take that into account when designing the clinical trials and to ensure diversity. Trust, I will take it more on the concept of the ethical side of things, because it is essential for increasing diversity, because over the years, underrepresented populations especially, but I find that you have that everywhere. People, because of historical, cultural, or systemic reasons, are skeptical of medical research. And that is everywhere. And so in order to be able to, and sometimes we can speculate what barriers are present in different places, but you can never be too sure. So you always have to keep an open mind there, and that is why there's a big push now to involve the patients and public in medical research, because you want to ensure that you are able to reach out to as many to the different populations and work against, not sort of working against the barriers, but work with the various people as much as possible. So, if you had looked at the abstract, there are many disease indications present. So what I want to do, take you through this journey, my career journey with you, and explore the multifactorial drivers of various diverse threatening conditions with case studies. So I have some genetic conditions I want to explore: sickle cell disease, Crohn's disease, cystic fibrosis, and also talk about, I'll take infectious disease. We can go forever talking about them, but I'll just take one example and talk about infectious disease. And then I will talk about sometimes the genetic diseases we encounter. There's impacts from infectious disease, which makes things more complicated. I'll touch a little bit on that. And then look at the consequences of underrepresentation and how either skewed data or ineffective treatments can be harmful to different populations. And then we'll discuss about how translational research itself has evolved from a unidirectional to a bidirectional, three-way model, and a multidimensional model. The unidirectional is the bench to bedside model, which is focused on applying scientific, historical, that's the first one, applying scientific discoveries directly into clinical settings. And then this moved on to a bidirectional model, as over time, people began to understand or realize the importance of having this feedback loop where you have the clinical observations informing laboratory research. Then the three-way model incorporates patients and the public to ensure that the research is relevant to the recipients of the research. And then we move on to the multidirectional model, which is, in that way, it's still the same three-dimensional model, but it's an emphasis of communication between all the parties involved. Now, this modern approach of a multidimensional model ensures that we have, it fosters a co-creation of knowledge rather than just knowledge transfer from the bench to the bedside, and helps us to have equitable research partnerships where patients, clinicians, and scientists can collaborate. And in this way, the public are able to trust science because they have made an input into whatever research, technology, or treatment has been produced, and they feel heard and included in the research process. Okay. So the first study, sickle cell disease, I will start by talking about this disease. This was the first research I did as I was a graduate student when I was doing, when I was working on sickle cell disease. And I will talk about it in terms of public health, homeostasis, evolution, and trust. Now, sickle cell disease is a hereditary blood disorder, which is characterized by abnormal hemoglobin, which causes the normal red blood cells to assume a rigid sickle shape. And it leads to blockages in blood flow. The symptoms include severe pain, anemia, organ damage, and increased risk of infection. This is a significant public health concern globally and poses a substantial burden on the healthcare system because of the high rates of hospitalization and the patients who really need and premature mortality, and they need care on a constant basis. Additionally, the disease impacts on quality of life, which makes it not only a clinical challenge but also a socioeconomic burden. And yeah, so how did we go to, So individuals with 2 copies of the gene, the sickle cell gene, develop sickle cell disease, and you have polymerization of the sickle cell leads to the formation of sickle cell fibers, and then you have severe health complications as a result. Now, evolution. This slide describes a paper from Nature back in 2010, where the authors looked at the associations between sickle cell gene and malaria endemicity. The red dots there represent the presence, and the blue dots the absence of the sickle cell gene. And the sickle cell disease itself occurs particularly in sub-Saharan Africa, India, and parts of Middle Asia, where the prevalence of malaria is high. The sickle cell traits, nature favored the evolution of that allele to be able to protect the individuals against malaria. So you see a direct correlation between how the endemicity of malaria in various regions directly proportional to the sickle cell, the intensity of sickle cell traits in those regions. And over the years, the medical interventions have taken to support this particular disease have been in 3 different categories. So we have the nutritional intervention, standard therapeutic approaches, and then, of course, the cutting-edge technologies, which hopefully we will see a reduction in the burden very soon. So, but our focus in the past decade was on nutritional intervention, which usually lead to a balance of biochemical metabolites in the bloodstream of the patients, of sickle cell patients, of the sickle cell patients. So, according to literature, as we said before, translational research evolved from a unidirectional model. That was the start. But then, and then that was during the early to late 1900s. It was mostly unidirectional. But I realized that when I look back and as a grad student and my professor who had been working on it for years, I saw that in this particular case, it was a bidirectional model. It didn't start in those in the regions where we worked. It didn't start as a unidirectional model at all, because here we were a group of local biochemists who knew how to use the local extract from foods to monitor and manage the patient's conditions. The number of patients usually exceed the hospitalization capacity that we had. So having biochemists who understood what to do with local means was beneficial for the patients. And they actually valued that support, and they were thriving with good quality of life despite their homozygous sickle cell trait. And in this case, we quickly moved from a bidirectional to a multidimensional model because more patients wanted to see this was completely non-invasive methods. They were using extracts which they knew about, and they could see exactly how we were measuring. My role then was to just measure calcium and phosphate levels in the patients, and that was all. And they could see how by taking those extracts, modifying the dose, how it helps to balance the biochemical metabolites in their body and how it's led to improving their conditions. So it was a great research program we had then, and it makes me feel that nature sort of has it all. And sometimes we need to look deep within ourselves and we can find answers that we are breaking our heads to look for. Take a bit of Yeah. So I'll move on to the second case study, infectious diseases. These are diseases caused by bacteria, viruses, parasites, or fungi. So I'll explore one example, and this is based on the work I have done under these four broad categories. In this case, when I was talking about sickle cell, I said we moved from a bidirectional to a multidimensional model. In this case, it was a stark unidirectional model. I mean, there was nothing to work with in this particular situation. But then it was also a success story because we did get to a bidirectional model, as I will show. This is I'm talking about the human African trypanosomiasis here, which not many people are familiar with because it is endemic only in sub-Saharan Africa and often fatal without treatment. Now, this particular parasite, part of its life cycle is in insects, and part of the life cycle is in man or cattle. Because there are three subspecies of the parasite. We have the parasite that affects the cattle is called Trypanosoma brucei, and it's amazing because it's really lethal to cattle, but it does nothing to human beings. And then we have Trypanosoma brucei gambiense, which occurs in the western part of Africa and causes the chronic form of the disease. Disease progression is slow, but eventually the patient dies. Trypanosoma brucei rhodesiense occurs in the eastern part of Africa. Disease progression in that is very fast, and patient leading to multi-organ failure and death. Before the recent interventions we've had on this disease, it was termed a neglected tropical disease with limited treatment options. Why I call this a stark unidirectional model was because we had drugs to treat the disease, but those drugs were actually prepared for other disease indications. The medical personnel there who only had to work with what was available to them. And so looking at the symptoms that the patients present and what is available matched a few drugs for the treatment of that disease. But it was a terrible situation because the patients who usually it just leads to death. I mean, anyone who contracted that disease, it's just a matter of time. And but the patients, as they die, they are dying either by the disease or because of the drugs, because the drugs were very toxic. And the WHO had to intervene quickly to see how can we remedy this situation. I was one of a few local scientists working on the disease at the time, and I had a bit of inspiration, sat down and designed a study how we can sort of have new drugs. Okay, the problem is that we do not have drugs against the disease. Now, there are two things we can do to eradicate this disease. Since the transmission it's transmitted via tsetse flies, and these are actually living in, very thick forests. That's where they thrive. So if you do some environmental work to ensure, that people are living in safer places, yes, that's one way. But also when people encounter the disease, what happens? So one of the things was that there were no drugs. So I sent my design to the WHO and said, I think with this, we can come up with some new drugs. We have the genomes of these parasites. We can actually design new drug targets. So the WHO sponsored the work, and that was where I was working in collaboration with some scientists from the Wellcome Sanger Institute and also from the Department of Pathology here in Cambridge. So I had two sites of research which came out of that study. On the one hand, this diagram is a tree which describes the relationship existing between all the eukaryotic organisms. These are organisms with a nucleus and multicellular, comprehensive multicellular architecture. Now, the blues there, that's where you have the animals, you have fungi and amoebozoa. The greens are the plants, and the reds are the excavates. That's where you have the trypanosome I'm talking about. And the orange color are the cell group, including epicomplexans. That's where you have the malaria parasites. So the reds and the oranges, they have very notorious organisms that we are always finding ways to solve the treatment strategies for them. But one thing which I noticed when I was working on this is that where we have the trypanosomes, the closest evolutionary related organisms to the trypanosomes are the bodonids. And they are free living organisms. They have no problems at all. They do not cause any disease. And the similar thing we found in malaria, where the epicomplex and the Plasmodium falciparum, their closest relatives, evolutionary, are the covarids, which are not pathogenic in any way. So I was interested to find out, or in our group, what was the difference? How did these organisms evolve for pathogenicity and non-pathogenicity? And what are the core proteins that are responsible for these differences? And that led us to, the legends here just shows different proteins that were expressed and found that they have amplified within the parasitic organisms, and some proteins that the parasitic organisms have lost the core function. And one of them, which we call DCP1, just the name of the protein, the capping protein, one of them we've actually, through a follow-up research led by one of the postdocs in the lab, then we found out that particular protein can also work as a drug target. So it was an interesting way to just look at how proteins, how the organisms have evolved and diverged, and insights. It provides insight for us in how to manage the treatment strategies. Now, this complex diagram, which looks like just a series of circles and lines, actually it's a network of all the biochemical reactions within the parasites. And the colored areas, the red and the green, are examples of two drug targets which I identified in the resource. And because there are very few scientists who are working on this disease, and immediately I identified this. My PI I was working with at the Wellcome Sanger Institute immediately called his colleagues in Belgium and Glasgow, because he said, look, we've done this job sort of blindly, just using the genome. But what we found out is that two of the drugs targets we have highlighted, you are currently working on them. So we think we should collaborate and bring in all the experimental evidence to see how we can sort of work together. So that was when we then wrote an application to the Royal Society and said, the WHO has sponsored this unique resource, and would you like to sponsor it so that we can make it an international portfolio and get everybody working together? Yes, so we got the funding for that. And then we built up on what we had done. So the idea, it was just a simple idea in terms of identifying the drug targets, where we looked at the unique and critical reactions that are occurring in the parasite, and we highlighted those critical reactions. The initial people who did this ran this algorithm in the US at Stanford, and then they called it choke point reactions. But then myself and my colleague here, we framed it to bottleneck reactions because we added some other things in the algorithm. So quickly moving on, we had an annual review of the situation in the past, I think it was a month ago, and the disease we have a decline of the disease in both West and East Africa. The number of reported cases have declined substantially. The most cases we have now are the Democratic Republic of Congo, and understandably, because usually when there is conflict, it's more difficult to address disease situations. But we now have 2 new drugs against the disease, and it's a success story, and we have moved quickly from a unidirectional to a multidirectional model. So now you have international and local scientists collaborating regarding this. Since we've seen progress in terms of treating the disease in humans, we're also working with the having interactions with cattle rearers and see how we could also support the disease in cattle. So this is a huge success story. Now, so taking one genetic disease, one infectious, now I'm going to another genetic disease, cystic fibrosis. I'm going to talk about cystic fibrosis in association with a bacteria. So cystic fibrosis is a genetic disorder caused by mutations in the CFTR gene, and it leads to the production of a thick, sticky mucus that obstructs the airways and digestive system and results in recurrent lung infections, respiratory failure, and reduced life expectancy. It is a serious public health concern, particularly in Europe, with significant impacts on the healthcare system because it is a chronic and progressive nature. But in this particular study, I will focus more on my contributions on evolutionary systems biology. Yes, okay. So another funny diagram with dots. And so this is actually the whole schematics of the processes in the bacteria, Mycobacterium abscessus. It gives this is a very important example of how a bacteria evolved from an environmental bacteria to an opportunistic one, and thriving in the thick mucus and inflamed airways of cystic fibrosis patients, where the antibiotic pressure and host immune system defenses drive adaptation. Now, the evolutionary adaptations by this bacteria makes it difficult to treat. It complicates the situation and leads to chronic and worsening lung function. So this was actually where I applied the refined methodology, where I changed it to bottleneck reactions and accounting for, because then when I worked on the human African trypanosomiasis, I only used the genome of the parasite to identify the drug targets. And then we had people who had done all the experimental evidences and then we brought everything together. But in this case, I had to account for not only using the genome of the organism, I had to account for the way the cellular metabolites are moving in the body, because we need to account for the quick adaptations of the bacteria within, you know, as it's thriving. What is making it to thrive in the host system? The host is fighting it constantly, but the bacteria is still thriving. So I had to account for how things are flowing within its system, and that's where we had that. So I have it colored between red and blue because at the bottom here, I said in association with CRISPR-Cas9 screens, was working, this is in collaboration with the floating lab at the Department of Medicine here, and another colleague was using CRISPR screens to look for essential reactions. So it was the same goal of trying to target the bacteria to be able to treat that bacteria in cystic fibrosis patients. But we came up with different methods and we had to see where our methods merged. And we used the methodology. Of course, it was interesting. We had similar, completely different methods, but we had similar drug targets and we used that to prioritize the drug targets. And what was a clinical observation generally is an imbalance in the metabolites in the cystic fibrosis patients, where the blue filled circles here demonstrate metabolic pathways with significant levels, and the plus sign accounts for elevated levels which are up to 300 and 500% compared to the other metabolites. Excuse me. The blank circles are indicative of depletive pathways. Similarly, the same thing happened here. So based on prior evidence and the changes that we see in the metabolites, we were able to, and this was also work done blindly because I was not in, and that's why I'm, you know, limiting my talk on this to just evolutionary system biology. I did not work directly with the patients. I only saw patient number 2, 3, 4, 5. I just saw that, but, you know, and this is an ongoing research. But based on the changes in the metabolites and what we see in literature, it's interesting that we're able to categorize them, the groups of patients, either early structural lung disease or resistance-related mutations. Now, the beauty of this is that you can actually take a patient's blood sample and indicate where exactly the patient is in terms of mild, moderate, or severe disease. And technology as it is right now gives a much deeper insight than we had before. And so when you are able to involve the patients and the public within this research, it helps you to be able to design a clinical trial that will be well-rounded and of benefits to everyone concerned. The Cystic Fibrosis Trust actually recommends involving people with cystic fibrosis as early as possible and throughout the design stage of the trial. I'll quickly move on to Crohn's disease. This is another genetic disease, but also has complications with infectious diseases. It's chronic inflammatory bowel disease affecting the gastrointestinal tract of the patients, and the symptoms include abdominal pain, diarrhea, weight loss, fatigue, and malnutrition. It is a growing health problem globally, particularly in industrialized nations, with rising incidence even in the developing regions, which are linked to diet, urbanization, and environmental factors. The patients usually require lifelong management, medications, and surgeries and hospitalizations, which impact severely on the health systems. And it also carries an increased risk of complications such as bowel obstruction and colorectal cancer. The association with infectious disease are potential triggers to the disease and usually lead to exacerbation of the disease condition. Now, this is a recent article which I highlighted here, which described the standard treatments that involve in treating Crohn's disease. And it is a bottom-top approach with options from mild to moderate to severe cases. Now, in this standard way of treating the patient, whichever treatment option you choose require a continuous assessment of the patient. When I was working on this disease, the literature which we published, we showed that in treating patients with moderate Crohn's disease, mostly using corticosteroids, we identified that if you use intravenous hydrocortisone on that group of patients, it was leading to a greater fall in blood potassium, and the patients end up having, the side effects were a lot, fatigue, diverse tissue and organ problems. And so we moved on to methylpred, which was not as, we didn't experience as much side effects for the patients. So, but we're moving on from standard traditional medicine to future precision medicine, whereby, because with standard traditional medicine, as you can see, whilst the goal, some patients, you achieve that goal of needing benefits for the patient with some patients, but some patients have no effect, and then for some, there are adverse effects. But what we're doing now, you have the clinical and molecular profiling of the patients, and then you can use that to predict what treatment would work best for which patient. And so when you have you have the genome of the patient, you also look at the expression, how the genes are expressed in disease conditions. You're able to match with different treatment levels. So with the ordinary eye, maybe someone coming into the hospital and the doctor might prescribe a sort of treatment for moderate disease. But you might be surprised that if you use the clinical and molecular profiling, you might end up giving that patient a different category of treatment. It might even be the mild disease treatment option that will be best for that patient. So that's clinical and molecular profiling does give a huge benefit. And how we engage the patients and the public to explain these diverse areas and how it will benefit the treatment is important. So the final one is on cancer, and quickly exploring in the light of the same categories such as public health, homeostasis, evolution, and trust. Cancer, as we all know, is a leading global health problem and responsible for nearly 10 million deaths annually, and incidents are rising due to aging populations, environmental factors, and lifestyle changes. Now, early cancer detection is critical for improving patient outcomes and reducing overall the global burden of cancer. The Alliance for Cancer Early Detection was set up in 2019 as part of a global initiatives to address the early detection of cancer. This alliance includes the Cancer Research UK and multiple institutions in the UK and the US. And this year, DFKZ, another institution in Germany, has joined the alliance. And the alliance is working towards not only looking at how we work with marginalized populations in the particular countries where these institutions are present, but how we can also work closely with low and middle income countries. There was a report recently, I think it was just sent out this month, actually from CRUK, so UK 2025 Socioeconomic Deprivation Report. It highlights that little has changed in progress since the initial report in 2020 and the report produced now. Little has changed towards tackling cancer inequalities. And the stark differences between the most and the deprived communities continue to be seen across the cancer pathway when you look at risk factors, diagnosis into survival and mortality. The report mentioned that depressingly, cancer-related deaths are almost 60% higher in the most deprived parts of the UK than in the least deprived areas. In addition, the burden of cancer is disproportionately higher in low and middle income countries. So there's a lot of work that needs to be done. And contributing to the alliance, we have the Cambridge East Clinic, which recruits healthy volunteers. It's led by Professor Rebecca Fitzgerald, and with a family history. And the clinic recruits healthy volunteers with a family history of cancer, but who have been free of the disease for over 1 year. Now, the patients receive very good support and counseling from our nursing team with a 95% annual retention recall. Now, the patients are able to join other studies. So annually, they come have their lifestyle questionnaires assessed and samples taken, have some few checkups. And sometimes if the profile matches or with other studies, they are welcome to join other studies. And we found out that a few patients, not many, a few patients in that category develop cancer during the period of the trial, and they are quickly moved to the hospital's patients pathway program. The clinic continues to welcome new participants, and we have the details on screen. So I have described how translational research has evolved over time from a unidirectional to a bidirectional and three-way model and a multi-dimensional model, and use different disease categories as examples to discuss the barriers under the 4 categories of public health: evolution, homeostasis, and trust. Now, there was a study a recent study sponsored by Novartis, where the authors were looking at the barriers for inclusive clinical trials, and included things like issues of miscommunication, mistrust, and finances, amongst others. And some of the potential solutions they highlighted included inclusivity strategies, proactive strategies, enabling better communications, co-productions, and trust is a very important element. I've spoken about how some of these barriers have been addressed through scientific advancements and ethical considerations. So health is an essential need for individuals and communities, and a multi-dimensional model that we have all worked towards, which involving medical practitioners and with the patients and public co-creating the treatment strategies is what we are aspiring towards for every disease condition. And so why do we develop these scientific advancements and work within the principles of evolution and homeostasis? Maintaining trust is an ethical responsibility that we owe to the public. And we usually do that to engage in the community, but language and culture shouldn't be a barrier. Seeing how we work with language and culture is important. The scriptures that usually come to me most times when I'm working in a new vicinity, interestingly, 1 Corinthians 15:31 says, I die daily. I die daily. It's like I'm going to a new community, a new environment. I forget everything I know. This is completely new. I'm open to the challenges. I'm open to see. I'm open. My ears are open. I'm listening. And there are some things which I will see here which I have not seen ever before. And I put them down, and and then 1 Peter 5:9 says, the same afflictions which are accomplished, which you are going through, are seen by your brethren all over the world. And that scripture all comes to me because sometimes, and this is not only with a disease indication, sometimes going through a particular circumstance can feel so overwhelming, and then you feel you are alone. But no, other people are going through that. And one very funny example with when I spoke about trypanosomiasis, and we sat down one day in a lab meeting talking about it, and invited a speaker from Ireland, deep in the Irish coast, to talk about trypanosomiasis. This is a disease that we have said occurs only in sub-Saharan Africa. And here is a man coming to tell us that they have encountered that disease in cattle, in farmland, that they have no record of movement between, that it was an isolated community in Ireland and no record of movement. And similarly, it talked about a particular community in Canada where they experienced the same thing. So sometimes never think that you're alone in something. It's happening in other places. Thank you.
Thank you so much. We've got some time for questions now. Can I invite people who are watching online, do add questions in the Q&A box. But anyone from the audience with a question?
Particularly interested in the slide you had on addressing inequalities, if you wouldn't mind putting that up. And the reason I'm asking is because I'm on steering a trial management group with Rebecca. And part of what we're finding in research that's just starting is the number of people from the lower socioeconomic areas are minimal. And we're wondering how we can access that, gain access to it.
Okay, so that is an ongoing issue. I know I was discussing with Sarah just now before I started the talk, because I mentioned to her some of the strategies that I am planning to use. And one of the reasons why I mentioned faith, because faith cuts across boundaries, cuts across cultures, cuts across barriers. So personally, one of the areas I'm looking at is to work with different religious communities and see how we can maybe get participation, engagement. First of all, it's engagement with these communities. So yes, we are looking at the same thing, but yeah, that's one of the areas. And through religious communities in general. There are various other ways, because if you go to different communities, there are community health centers in the different communities. So engaging with those community health centers could also help.
I can articulate this. Can you share anything about how your faith is helpful or comes out or is seen as appropriate to share or be part of your work in the different places where you worked or the different communities you work with? Because I imagine the dynamics must be really different in the different places.
That is true. The dynamics are different, but the principles are the same. What I noticed, and I think I got educated on that from my daughter doing religious studies and looking at other religions. There are lots of similarities in terms of the principles of spirituality. And so I don't approach it as, because I have a background of the Christian faith. But if I'm going to a Muslim environment, I don't go telling them what the Bible says here or the Bible says there. I allow the principles or the teachings of my faith to speak in terms of my approach to the community. And so I use the example of I die daily. So I'm not going to say that to them, but it just reflects in my approach of being open towards what is going on in the community. What can I learn from you? And see how we can engage with one another better. I don't know.
Thank you for your talk. I was wondering if kind of the main funding bodies of clinical trials that there are some main funding bodies. Do they share this exact same approach worldwide? The kind of idea that diversity is important in trials, is that well accepted and implemented across funding bodies?
Yeah, in fact, that is the message, the core message across most funding bodies now. CRUK, Cancer Research UK, the founder for this, the UK institutions in the alliance, in fact, that is the major message they've given to all of us working on this, that we have to involve patients and public. And I also noticed that different funding bodies in the UK also are applying the same strategy. Everyone wants to see how we are involving the patients and public in our research.
Can I ask a question? So you've talked briefly about barriers to inclusivity in clinical trials and ways to overcome those barriers. Can you give more specific examples of where you've seen barriers, whether it's trust or communication, and how you've seen those barriers overcome?
Thank you. Okay, so whilst I was working that was one slide I took out eventually from the talk because it was too much. When I did I sort of led a global study that was sponsored by Pfizer on antimicrobial resistance across different continents. And so I was responsible to ensure that we had recruitment. We had a target recruitment from different countries and continents. So we had in Europe, we had US, we had in Asia. So we just took one country in each continent to work with. Yes, so that was a huge challenge. And so what I did there was that I went in, because over the years, I've worked a lot in diversity, ensuring diversity, addressing inequalities in health and education. So I seem to have contacts in various countries and continents. So I reached out in Asia, we were working with people we wanted to recruit from India. So I reached out to my contacts in India. Unfortunately, I had someone who was in the student in the medical profession. And so we recruited him to work with us. Yes, we recruited 2 people in India to work with us within the community. One of them is Indian, one of them is Sierra Leonean, who happened to be in India and speaks the language. So they were able to move in with the hospitals and work with the community to achieve the targets. So by working with the community, I was able to, so it was an online strategy for me. The only country I visited during that period was Spain. I couldn't find someone. Spain was the European country we chose to work with. And I couldn't find someone who could work with me in Spain. So I had to go there myself. And when I went to Spain, you can get by with English and everything, but it was the first time I had to bring Duolingo out and started learning some Spanish. Yeah, and language is when you identify. For me, identifying with someone's language, it goes a long way. Immediately, I went to the hospital. You wouldn't even know I didn't speak Spanish. Maybe my accent would have given me out. But the fact that I was able to greet in Spanish, I was able to ask the main questions I wanted to ask in Spanish, opened the doors for me. And I was able to, mingle through. And it was great, because I would say within 6 months, I established relationships in every country we were working with. So, yeah.
So it sounds like relationships are very important.
Yes.
Any further questions? If not, we would like to thank you once again for a fascinating talk.
Thank you.